Hey guys, this is Kevin Budiszewski, and these are the fluid compartments. So the entire body is made up of two things, water and non-water mass. Water mass accounts for about 60% of the total body weight, whereas non-water mass accounts for only about 40% of it. The total body water is split up into two different compartments, intracellular fluid, which accounts for about 2/3 of the total body water, and extracellular fluid, which is a smaller component making up only about 1/3 of the total body water. Do you guys know which electrolytes make up the intracellular fluid? Well, the big one that you guys should remember would be potassium. What about the extracellular? It would be primarily sodium and chloride. Good. The extracellular fluid also houses the bulk of albumin and bicarb, whereas the intracellular also houses a lot of ATP. This should make intuitive sense, as intracellularly, we need the ATP in order for that cell to do work. Do you guys recall where the bulk of calcium is? That would be outside of the cell. This should make intuitive sense, and it's a very important pathology tie-in. So what do excess amounts of calcium trigger? That would be apoptosis once calcium is inside of the cell. We'll enforce these relationships further at the end of this video. Extracellular fluid can also be divided into two components, interstitial fluid and plasma. Interstitial fluid makes up about 3/4 of the ECF, whereas the plasma makes up only about 1/4. It's intuitive that the interstitial volume would be bigger because that's the volume that actually surrounds our cells, whereas the plasma volume, on the other hand, is only the volume contained within the vascular system. So this is a lot of information, but an easy way to remember it is the 60-40-20 rule. This means that 60% of the total body weight is water. The 40 means that 40% of the total body weight is intracellular fluid, and the 20 means that about 20% of the total body weight is extracellular. Which, if you think about it, these percentages should make sense because 2/3 of 60% is 40%, and 1/3 of 60% is 20%. For your test, it's also important to know the various substances that can serve as surrogate markers for these different compartments. As it turns out, the volume of extracellular fluid can be measured by inulin or mannitol. The reason for this is that these molecules can move out of the vasculature but are not taken up by cells. Plasma and all plasma volume can be measured by radio-labeled albumin for the opposite reason. This is because albumin protein is too large to pass through the capillaries out of the vasculature, so it's an excellent surrogate for the plasma. Albumin is responsible for the creation of the plasma oncotic pressure as well. Do you guys remember what this is? It's the pressure created by proteins, notably albumin, that pulls water into the vasculature. Remember that this is a pulling pressure and not a pushing pressure. Albumin's too big to pass through the capillaries, so we should never see it in the urine. However, there are times when we will see protein in the urine for pathologic reasons. Can you guys recall a syndrome in which proteinuria is typically seen? That would be in nephrotic syndrome. These patients typically present with protein in the urine and a decreased plasma oncotic pressure because all of that albumin is being lost in the urine. Therefore, we don't have a pulling pressure. Alright, so just to solidify some of these relationships, let's walk through a sample problem. So how is the body water of a 70-kilogram man distributed? Let's walk through it together. If he weighs 70 kilograms, then 60% of that will be water, which is about 42 liters. 2/3 of that 42 liters is intracellular, which would be 28 liters. The remaining 1/3, or about 14 liters, is extracellular. Of the ECF, 1/4 of it is plasma volume, and 1/4 of 14 would be about 3.5. And the remaining 3/4, or about 10.5 liters, would be interstitial. Be sure to keep in mind the 60-40-20 rule, and these calculations will flow a lot easier come test day. So let's talk more in depth about fluid compartments and what they're actually made of. The major cation in the intracellular fluid is potassium, while the major cation in the extracellular fluid is sodium. Some people remember this by thinking of it as a banana in the open sea. Think of all of that potassium surrounded by salt water or sodium chloride, and you'll never forget these major cations. Potassium's on the inside and sodium and chloride are on the outside. So these two cations have established concentration gradients that encourage movement from a high concentration into a low concentration. So let's show this by example. Here is a very high-resolution image of a cardiac myocyte, where depolarization of the cell membrane occurs when the voltage-gated sodium channels open, allowing sodium to enter cells. This occurs down the sodium concentration gradient, causing muscle contraction. The cell will then repolarize once the voltage-gated potassium channels open, allowing potassium to exit the cell down its concentration gradient. The ECF and ICF also have different anions. In the ICF, it's mostly protein and organic phosphates, where the majority of the body's proteins and ATP are located inside of cells. In the ECF, the major anions are chloride and bicarb. Remember that bicarbonate is used as an extracellular buffer and a means to transport carbon dioxide. Alright, time for a flash quiz. What is the 60-40-20 rule of the body's water compartments? The 60-40-20 rule means that 60% of the total body weight is water, 40% of the total body weight is inside of cells, and 20% of the total body weight is outside of cells. As a bonus question, what fluid compartment can arenulin or mannitol be used as a reference for? Correct, that would be the ECF.